Mining Gene Plant Trait Association for Rice by Gene Trait NER, Ontology Merge and Ontology Annotation. We will introduce our work from 4 aspects: background, method, performance evaluation, conclusion, and future plan. First, background. Our project aims to mine gene rice trait association. As we all know, gene trait association is of utmost importance for rice cultivation and molecular breeding. But now, rice trait mining still lacks method and corpus. One main difficulty of automatically mining the rice trait from text is that the description of rice trait is virus. Let's give an example of strice trait. In our early research, we found a couple of similar work, TAS and fungus gene. Let's give a brief overview of them. The TAS used a manually curated way, while fungus gene combined a manual curation and automatic matching way. More details of their work are shown as a table. We did an artificial quality check on these two work. Both of them are meaningful and inspiring, but of course there is room for improvement of their results. Next, we will introduce the plant trait ontology. It was used in the project TAS we just mentioned. And the information concerned is the class ID, preferred label, synonyms, definitions, obsolete or not of the rice trait. Based on this data, we designed the flowchart of our project as the figure shows. Next, our method. The first one is gene NER. A mature gene tagged into a pulpator has a low recall rate for S, so we use HUNER instead. For a text with a title and abstract information, fungus gene summarizes a normalized gene mention from the text, while HUNER extracts all kinds of gene mentions rather than a gene symbol. Second, the ontology merge. In order to get a more comprehensive rice trait ontology, we found several ontologies that may contain available rice trait term, as the table shows. We now just merge PTO and WTO because PTO is a standard plant trait ontology, and WTO contains environment conditions and some possible rice traits. Later we may merge more ontologies. The process of merging ontologies as the figure shows, and the merge rules we observed are also shown in this slide. Next, we will extract the trait phrase mentions from all PubMed literatures related to rice. We use the pattern matching method and embedding-based phrase extraction, which we call EBPE, to improve the results. Details of EBPE are shown in this slide. Next, we need to map rice trait mentions to ontology ID provided by our merged ontology. Just like this figure, the left column is the trait mentions we extracted. The right column is the normalized term in the rice trait ontology. In the beginning, we manually construct this training set. Later, we will use BERT-based model to automatically complete this classification task. But this is not a simple task. The possible difficulties are shown as follows. In order to better realize the function of automatically mining the rice traits, the ongoing work ontology annotation is important. It will serve as a corpus and training data to a further supervised learning method. Next, we will introduce results. First, about gene NER. It can extract more gene mentions than fungus genes, but HUNER just extracts gene mention without corresponding gene symbol or ID. And you can see in the left column of this fig, it shows that the gene mention that HUNER extract is a virus. So we need the ORISA base to do the gene normalization to get the gene symbol, and then map a RAP ID or MSU ID. But many gene symbols in this database lack the ID, so we intend to use Pyrus, a Python package, to solve the problem. Second, the result of our EBPE method. We made a great progress both in quality and quantity, because our rice trait phrases are obtained through the cosine similarity calculation in EBPE. Third, the gene rice trait association result. Our gene rice trait pairs in the same sentence are less than fungus genes. One possible reason is that the keyword of fungus gene is not so rigorous. The rice trait, like plant and protein, means not all of the gene rice trait pairs in fungus gene are available. And we also evaluate our work from two aspects: merge ontology and extract the gene trait pairs based on the abstracts. Compared to the fungus gene result, our method performed better, and we manually check all of our results. They are correct and convincing. In the future plan, we are writing a Python or R package to realize the enrichment analysis of rice gene and trait term. just like geo-enrichment analysis. Details and the function of these packages are shown in this slide, and here is an example of our package. At last is the conclusion. There are still problems existing in gene normalization and ontology merge in the current progress. And after completing the 2.1,000 project annotation, we will evaluate our work from 3 aspects.